in India working on synthetic biology and its application in health, energy, and environment. Currently, he is the head of synthetic biology in lab at the School of Biotechnology, Jawaharlal Nehru University in New Delhi. Today, he'll be talking about the need for this new field, its foundational principles, methods and tools, and industrial applications, followed by a discussion on his work on the non-expression genome and its potential applications. Without further ado, I'd like to welcome Professor Dhar on behalf of all of us present here. Professor Dhar, the floor is yours.
Thank you very much, Vidisha, and good evening, everybody. And it's wonderful to be on this platform. I remember when I came back to India back in 2010, we had a trouble finding who is interested in synthetic biology. So then there was a first conference of synthetic biology. At the time I was in Kerala, but I'm so happy that now we are looking at large numbers and a lot of interest. So I'm sure that in the future, synthetic biology will not be new to people. However, there is still a concern because when I talk to many people, both faculty and students, and we talk about synthetic biology, people seem to know what is synthetic biology, but when it comes to definition, there seems to be a large variety of definitions. Some people also say that it is biotechnology renamed, or bioinformatics and chemistry, physics, a combination of that. So my humble attempt today will be to describe my view of what is synthetic biology. And the reason why I'm talking about conceptual foundation is because my introduction to synthetic biology happened through the first synthetic biology meeting at MIT back in June 2004. This was the first meeting of synthetic biology, and I was very fortunate to be there, where the foundational principles were discussed and the need and the best practices. So I will be bringing forward learned from this meeting, and also what has happened in the community for the last decade, a little more than a decade. So to begin with, may I request everyone to please put yourself at the mute, and if you have any question or any comment, please interrupt me and let's talk about that. Or if you want to reserve your observations towards the end, it's all right with me. So I'll be switching off my video to make sure that the bandwidth is taken care, and I'll share the slides now. Just give me a second. I'm trying to share my slides. I hope you can see that very soon. Are the slides visible? Anybody?
Yes, sir, they are visible.
Is it okay? All right. Thank you very much.
Yeah.
So once again, thank you very much for inviting me, and I would like to finish my presentation in 40 to 45 minutes, and then we can have discussions. So the reason why we talk of synthetic biology is that, in fact, I'll be covering my talk in 3 sections. First, the fundamental concepts, then the Indian aspects. I'm not covering all of that because the time limitation, and then our project. So the way science has been organized is by investigating the higher structures and trying to see if an organism is functioning, what is the reason for that? What are the structural correlates that can map to a certain function? So the quest to answer this question way back from Mendel's time, when he imagined something, he did not see that, but he imagined something. And he zoomed in and zoomed out, and we call them as factors, genotype, phenotype, and so on. The whole idea was to find out what an organism is made of so as to understand the behavior of the organism. It can be as simple as a cell or as complex as a eukaryote. But eventually, the whole quest is to go deeper and deeper and try to see the structure. So when you look at this structure at the systems level, automatically the concern is what next? So then people figured out that the system was nothing but a collection of organs. Now, when people wanted to see the pattern of the organisms, their connectivity and their attachment with the muscle, bone, the blood vessels, nerves, cartilage, and so on, more and more details started coming in. And with the advent of technology, the emergence of microscopy, people started going in more details. So from there, people went to the level of cells. And in the beginning, the cells looked very innocent, but as the staining technique and the microscopy got more sophisticated, more and more details started emerging. And chromosomes and DNA became the superstar back in 40s and 50s, obviously because of its genetic transmission from the parents to offspring. So at that time, DNA was considered more at the regulatory level, that this is the big guy who's doing everything. However, over a period of time, RNA and protein community caught up and they proved that no, it's not DNA only, but RNA and protein equally has a big role to play, in fact, a much bigger role. So DNA became more of a database in conceptual understanding of scientists. So when you go beyond DNA, what you end up is chemistry. This is nothing but chemistry. Here we just study the molecules as a function of their chemical behavior. So you have the bond angles, you have the rotation, you have torque, and all kinds of things that you learn in chemistry. But because it happens in biology, we call it biochemistry. When you go one step deeper, you come across the fundamental physics level. And so now, if you go a little deeper, you need a equipment to understand the space, because 99% of atom is space. So it's better to zoom out and stay at the level where our analytical equipments can help us understand the structures, right? So this entire process is called reductionism. And why I'm talking about reductionism, not a synthetic biology, I'll come to that very soon. So through reductionism, using different technologies, we got the data. So let's say from the cells, we got genes, RNA, proteins, basically fundamental components. Now, when the Human Genome Project took off in a very big way, there was a bigger problem, that was how to deal with this data. Now you have a lot of data. Earlier, you didn't have so much of data, so we could manually think of documenting it. But now you have a lot of data. What do? You make use of computers, you make use of databases and analytical algorithms that can analyze this data, that can display the data or the results, and that can manage the data, keep it secure and all that. So the era of databases and modeling came into picture. At the higher level, you had pathway models. At the lower level, you had single component models like genes, RNA, protein, and so on. So now this was a phase of integrating the data. So reductionism was very successful, because of which you got data. Now integration is successful, because of which you got virtual models. And you capture the data. Now scientists, a little before 2000, a couple of years before 2000, they asked, how about the third approach in science? Now, these two approaches have been very successful. How about the third approach? And what was that? That was engineering systems, constructing systems. So they said, well, we are good in reductionism. We've been highly successful in capturing the data and modeling the data. Now, let us think of constructing the systems from scratch as an engineering approach. So the first approach is more popularly called as molecular biology and biochemistry, and the second one, bioinformatics and systems biology, depending on the scale at which you're looking. And the third one is called synthetic biology. This is very, very important for people to understand, because otherwise, you think of synthetic biology as something like synthesis, chemical synthesis. And some people say, oh, metabolic engineering is synthetic biology. Somebody will say making new DNA or hacking the genetic code is synthetic biology. Please understand that the whole idea of proposing this name, and this name, let me also clarify that on the third day of the event, June 2004, first synthetic biology meeting at MIT, Drivendi opened up this question to all of us in the audience that if you do not like synthetic biology name, please propose a new one. So people started thinking of, and there was a huge debate because people did not like this term synthetic biology. They said, it is a misnomer, it's misleading. If it's engineering approach, then there should be some other terminology. So then Drivendi proposed constructive biology as another terminology after a couple of months of heated debate. People said, no, it cannot be that because you're trying to imply that the rest of the biology is destructive. So he said, okay, let's call it, somebody said, okay, let's call it biological technology. Then what is biotechnology? Okay, let's call it biosystems engineering. Then you're leaving the parts, you're only focusing on the systems. So likewise, people kept on thinking of new names. And finally, the community sort of agreed for this term called biological engineering. So if you read papers on biological engineering, they're exactly the same in terms of its concept as you find in synthetic biology. So there's no difference between these two. It's just that your preference matters. When you talk of capturing the data and taking more of a collective behavior, because you can design a system only when you know how a system operates. So it's very important for the systems biology to help in the synthetic biology community to tell how this, and this was the first virtual blood cell, which was released back in 1998 from a Japanese group. And then you have people capturing data at different levels. So I proposed some time back, well, we have a lot of data from the reductionist approach. So is this a time to construct a periodic table in biology? And if yes, what would be the property of such a periodic table? Now, why we are talking about these things is because we are talking of engineering biological systems. When you're engineering, you need to get constants in hand. If you do not have a constant, if something is varying all the time, how do you engineer? Because you create a strain and you see that, okay, in the lab, today the strain is behaving as it is told, but maybe 1 week later, you're losing the control. Now, that's not engineering. You're moving towards engineering, but that's not engineering. Engineering means it has to be constant. So we have to find constants at different levels. The key point here is really that whether biologists can think like engineers. Now, if you're talking of engineering and biology in the same breath, then we look at similarities between two, as you can see on the screen. However, there are also dissimilarities. Well, there are some of them. There are more, actually. You have mobile components. Now, this is very important to understand because people are excited about creating apps. Okay, I'm installing a genetic app. I'm creating a logic gate. I'm creating a truth table. All of this is fine. It looks very nice, but in the real practice, it doesn't work like that. Because the motherboard in case of computers has anchored components. Here you're talking of RNA and protein that are moving all the time and you don't have any control over their mobility within the cell. And biological systems are predominantly analog, but that would mean let's say, you switch on the light and it's on and off, but in case of fan regulator, it is 0, 1, 2, 3, 4, 5. Now this is analog behavior. That is how genes behave. And the other things. So when you construct something, you have to deal with a system which is noisy. That means the expression levels go up and down all the time. So you cannot catch a point in the graph and say, this is my gene expression. No, it's a pattern. It's not just one point. And also it's unpredictable because you're dealing with nonlinear systems, right? So when you construct something in engineering, you control the flow of information, but to even create one switch, you have to go one level down. You have to go from gene to RNA to protein and then hope that this transcription factor will activate the next event. But actually, it's more complex than this. Well, you also have to worry about the degradation at both the steps because most of it gets degraded anyway. So if you have enough RNA, and enough protein, and it happens to be in the neighborhood, and the molecular crowding effects don't mess up the design, then the next event will be switched on. Okay, so it's not as simple as electronic circuit designing, and that's the best part, because once you design something, you feel great about it because you've overcome a lot of issues in the system. So to design well, we have system one in standards and rules of compositions, and this is what we learned from engineering community. Standards are fixed quantities that do not change, that need to be fixed, whether they are naturally fixed or artificially fixed, but you need to constrain them. And the rules of composition means in which order do you combine the components in order to create a module, right? So you need both of them. IEEE standards do not exist in biology because organisms were not created to solve human problems. These are human problems, right? So it's good to use engineering as a philosophy, as an approach, instead of copying, pasting equations from the engineering textbooks or using engineering tools, thinking that if I put my gene expression data, now there is an engineering tool, I can model, construct, and deploy the system. That's not going to happen, because systems are totally different in their original nature. Absolute engineering solutions do not exist for biology because probably engineers did not have an opportunity to encounter a complex problem as complex as biology. This is not to downplay the contribution of engineers, because if you look at Boeing's 747 aeroplane, this is 7 million parts and hundreds of kilometers of wires. It's a mega complex structure, flying in the sky. But we are talking of an E. coli, which is more complex than a Boeing aeroplane. Here we are talking of complexity as in the emergence, which means that when the components are together, something new happens which you didn't know before, right? Probability, there is no guarantee that you make the RNA and the protein and the next event will be on. It's just a probability that it may be on. There's a likelihood that it will be on. And of course, the concentration gradients, as you know, during development, they play a role. So we have been good in decomposing the system one part at a time. Now we are trying to compose the system. And that is where I'm trying to put more emphasis on the fundamental aspects of synthetic biology for everyone to connect with what was the original idea of starting it. It was not a chemical synthesis or organic synthesis or something like that. That can be an application, I agree, but the original intent was to really engineer something in a controlled way. So the biotruth table concept was proposed June 2004 in this meeting. And then there were a couple of PhD students in MIT who were given these topics, and they could barely finish, creating a digital switch out of an analog system. And this did not go well, because they thought that if we create, let's say in E. coli, you create 4,000 truth tables. In yeast, you create 6,000 truth tables, right? You can construct the genome and you can, redesign it, but it doesn't happen that way, because you're talking of two different systems totally. Well, we can, feel comfortable with the analogy, but really one has to understand that when you're talking of rational design and controlled construction leading to applications, you're talking of an engineering. Now, why are we talking of engineering again and again, given that there is already genetic engineering? So why am I talking of biological engineering now? You see, genetic engineering was a mission. It was a vision. We never engineered genetics, right? So we use the word genetic engineering, but we never engineered anything. What we do is we have thousands of these constructs in the hand, and we throw them on millions of cells swimming in the petri dish or the multivale plate, and some of them through electroporation happen to go into the cells and express themselves. So you get 8 colonies, 10 colonies, or whatever. And if you're very lucky, you get the orientation at the right place, right insert, and everything is fine. But that's probability. That's not engineering. So though we are saying engineering, what we are actually practicing is probability. So engineering is started for the first time. Not that we have solved the problem, but the intent and the approach is very clear. Whether it will be successful or not, we do not know. Nevertheless, companies are using organisms to commercially produce certain substances, and you can see the payoffs are good so far. It has not picked up in a major way, but still we have the proof of the concept, which is good enough to scale up in future. So that was about introducing synthetic biology from the fundamental concepts perspective. Now, how are we doing in India? I'm not capturing all the science that we are doing, excellent science that all of you are doing, because that will be too much for this meeting. I just tried to capture the headlines, the way it has happened. So the IGM started in June, somewhere maybe October or November 2004, and I think the date was fixed around November or December. And ever since it has been happening around the same time. Earlier there were 5 teams and 31 participants. But now over a period of time, I've not captured 2021 data, which is not available from the team perspective, but from 2004 to 2020, you can see from 5 teams it has become 362, and it was little more earlier last year, and 4,800 participants, right? So it is a huge jump. From the Indian perspective, the first team that was sent to IGM was by Mukund Thattai from NCBS. In fact, the following year, he again sent the team along with the mentorship of Guhan Jayaraman from IIT Madras. And after that, the pace has picked up, and DBT started funding this, couple of years before the IBEC initiative was started. They were funding it in a different form, but then we proposed that this has to be formalized, because we are only serving the foreign community all the time. So we need to have some initiative ourselves, so that we have the competence, we have the money. Why can't we have a competition? So because at the time the budget was an issue, and so I had a discussion with the current secretary. So she agreed that we will call it IBEC, that means Indian Biological Engineering Competition. However, it will be a kind of a pre-IGM, wherein students are, when the applications are screened, and then people are supported up to a maximum of 10 lakh. And then per team, of course, that will not be enough of a money to participate in this event. But nevertheless, there was something which was, beginning to shape up. As you can see, over a period of time, more than 50,000 people have participated. And from India, more than 1,100 people have participated in this. More people were supposed to participate, but then they withdraw the application at the last minute because of financial or other reasons. If you look at the maths, so far 71 teams have participated. Of course, the same team have come many times over, so it's kind of an overlap here. If you consider the current registration fee, which is around 5,500, now if you say that, okay, earlier it was not so much, now it is little more. If you take an average of 4,000 per team, that means from India, we have spent 2 crores on this. And if you take travel and accommodation into consideration, the total money that has been spent will be far more than what it looks like. So with this money, I believe we can organize we have the money. It's not that we don't have the money, but it's just scattered, and it has not come collectively together. The moment we are collectively on the same page in a formal setting, then it will not be difficult for the money to come in. It is just that we didn't have the right numbers, but I'm happy that with more than 1,000 people or from India going to IGM, and I'm sure many more talking about IGM, we have the right numbers now to launch bigger initiatives. So the first event that was organized was Biodesign India. That was back in October 2010. And there was a voting that what do we call this new field, or internally, what do we call it, so that we start with the mailing list, then put a website and all that. So Ashwani, who was from ISC Bangalore, she was the student of Nagasuma Chandra. Professor Nagasuma Chandra was also speaker in this event. And I had to literally Google to find out who has interest. We're not talking about work, but interest in the group. So I invited people from IITs and central universities and so on. And finally, we could put together the 3-day event. And then we asked participants to name the mailing list or the website that might come in future. And so she proposed Sanjeevani, and it was accepted by most of the participants. And we have a 100+ mailing list there. And the second major event was organized in JNU. And here we had a joint Indo-US program. And the best part was that in this event, many students got PhD promises by the speakers from the US. So that was really, really good as an outcome. And the third event we organized in 2007 was what we call as IBM, International Biological Engineering Meeting. Here also we had people coming from Hopkins and other UCLA. So and of course our own Indian community. So it was a really major event, and several topics were discussed. But in future, we really want to continue this as a series. So I will really look for your help in organizing this. And if you have another name in mind or another setting in mind, do not hesitate to discuss with me. From our own small contribution level, when the field was launched back in June 2004, I spoke with Ron Weiss on the third day, and he had a talk. Ron Weiss was from Princeton at that time before he joined MIT. I said, look, now the field has been launched today and we don't have a journal. So he said, okay, let's do this together. And so we started this Systems in Synthetic Biology journal with Springer. We could barely manage to run it for 8 years. Because what happened is during that time, many established journals opened up synthetic biology sections in their journal. So we lost the market. And, there were more heavyweights, and Nature and PLOS and now ACS, all of them came up with. So obviously we had a tough time because the community was small. If it was a big one, then probably we would have been able to survive. But then we thought of closing it and merging it with the Springer other journals. And 2018-17, actually, I proposed this to DBT that we need to train our students in synthetic biology from remotest corners of India, and we successfully accomplished this for 3 years. We had trained 45 students, and we hope that some sensitivity and sensitization of this field has happened. But when I look at the numbers, the IGM numbers, I'm so delighted that, it's a huge, support to the field, when you're involved. So now I'll come to the project that we are doing. And this is what we call as the Dark Genome Project. And I will very soon tell you what is the reason why we call it dark genome. So you see, traditionally by converting gene into junk and garbage, we have learned biology. And Susumu Ono said long back in 1925 in an article, there's a lot of junk in the genome. So we said, okay, now this junk, previously known as junk, is now known as RNA coding gene. But still there is some amount of genome that does not seem to express. It's truly junk in that sense. Susumu Ono's perception of junk DNA has been addressed to a great extent when the RNA community came up with the RNA coding genes, the non-coding genes. It is not non-coding, it is coding for something, but only RNA. And the bulk of the genome codes for RNA. It does not go to the level of proteins. And some amount of genome neither encodes RNA nor proteins. And we are after this, region of the genome that we call as dark matter. This also emerges from a simple observation or the question, why nature placed signals at specific spots in the genome as start and stop signals? Did she do all kinds of experiments and then said, okay, the ones that have qualified the exam, they become protein coding genes. The ones that have not qualified are trashed, or we don't install the signals there any longer, and we retire the genes over time. Well, retiring of the genes over time has happened, and we call them as pseudogenes. However, we do not know if nature performed all kinds of experiments. If that is true, then technically we can make more genes. Even from the ORF, you can make more genes. From ORF to intergenic sequences, overlapping, non-overlapping, you can make a lot of genes, right? Theoretically speaking, I'm saying. But when you talk of that, we have to be very careful. What kind of sequence are we talking about? So back in 2007, when I was with Riken in Yokohama, we initiated this experiment and we considered E. coli with intergenic sequences that have no transcriptional history. So we took 2,500 E. coli intergenic sequences, we converted them into the protein sequence equivalent, matched against the NCBI NR database, non-redundant protein database. Wherever we are getting the sequence similarity, we are discarding the sequence. Wherever we have absolutely no similarity, we take that sequence. But not only that, but we also say that if it is conserved, we discard that. That means if it is not matching the NR database, but it is conserved, we also discard that. So 2 filters basically. After that, we came up with some sequences, and you can see the red colored lines on the screen, all randomized, overlapping, non-overlapping, different directions, small and big. We did not know if it will work. So the odds against these ideas were many. This was the first such attempt, and there are other things. When I, after this initial bioinformatics, when I, took the project to my students, PhD students and postdocs, and I said, this is something that I would like you to think about. Normally, they don't agree with each other, but for this, all of them agreed not to do the project. They were not interested because they said there's no previous publication, so if I fail after 2 years or 3 years, then who will take the blame? So it's a big hit. And if I have to do 2 projects simultaneously, I don't have time for that. So sorry, we cannot do this. We respected their opinion, and I hired a person from Singapore by name Chosu as a lab technician. She was not trained in molecular biology, but we trained her in molecular biology, and she did all the experiments. And when she got results, a lot of people jumped in, and to honor her, the first publication, she's a co-author on the paper. Normally, you don't have lab technician as the co-author, but then we did respect her because this was a turning point in our research. So, how do you make a gene out of non-expressing DNA? This is very simple. It's a tiny problem because you know how a gene looks like in the textbook. So, logically, you put signals upstream and downstream, and you have made a gene theoretically. But if you work at the genome sequence level, it's a lot of effort because there can be gaps left between the signals, there can be mutations, and the signals may not attach in the right orientation. Anything can happen. So we said, let's make them outside the genome. What we wanted to do was to design a vector that will supply the signals, the upstream signals and downstream signals will be provided by the vector, and only the insert will be provided by the user. While we were wanting to design a vector, we were pleasantly surprised that Invitrogen had released the same vector around the same time, but probably for different reasons, we don't know what was the motivation behind that. But this was like a godsend for us because our 1 year of effort was, saved. So all we have to do is take the bioinformatics predictions that you get from bioinformatics, and take the bioinformatics. Sorry. Hello. Am I audible?
Yes, sir, you're audible.
Okay. Because sometimes the voice goes away, so I want to be sure. All we have to do is take the predictions from bioinformatics and amplify that, insert it, express it, and there you go. So our best moment came when we found that in presence of inducer, the gene is expressing. In absence of inducer, there is no expression. And so this was a major excitement for us. And now the nature of question changed. The first question was, how are they looking like? The proteins, what is the structure? So in our campus, we had 42 NMR machines. This was the largest collection of machines in Japan because, we were part of the Protein 3000 mega, challenge project. So our scientists, the Reikan scientists, have to solve 2,500 structures in 5 years. Rest of the Japan, 500 structures in the same duration. So the machines were running absolutely full. There was no slot available. So we have to go for a computational prediction. This was done at the Bioinformatics Institute of Singapore, where I was working before, and I requested my colleagues to help me in that. And they said that, look, out of 6 proteins, 2 of them look like they have a good structure, so you can go forward. And now, obviously, the question is, what are these guys doing in the cell? So when we were expressing the proteins, we found that out of 6 proteins, so we took 10 samples, and we lost 4 of them, and only 6 survived. And one of them, we found that when the gene is expressing, within 2 hours, the numbers are coming down. The cell numbers are coming down. So we said, if this is true, if this is the true positive, and we did this many times, I think 5 to 6 times, then if you switch off the gene, the growth should get restored. And that's exactly what happened, as you can see on the graph. The yellow patch, that's where we stop the gene from functioning. And we, when you switch off the gene, the cell growth is going up again, thereby proving there's a linear correlation. However, the story is not so simple because, remember that we did not design the vector. We took the vector commercially available. So the vector was not absolutely helping us to attach the insert right after the start codon. There was an overhang between start codon and the site of insertion. So that means we have to live with this because we are not removed it, we have to live with it, which means the results that you're observing here, they are not exactly because of ECA1. They're ECA1 plus upstream vector and the part of the downstream portion of the vector. 3 aspects coming together to create the phenotype. So when we noticed this and we found that the vector was contributing a lot, we said, let's change the strategy totally. And here we took the PET expression vector and we changed the strategy, because of which we took ECA3, which had no growth inhibitory effects. And when we changed the strategy, we found that there is 0% vector contribution. So earlier there was no growth inhibition, but now the cells were growing 3 times slower. We were again very lucky to catch this phenotype. If the cells would not have grown slowly, we at that time had no other way to confirm the effect of producing intergenic proteins. Okay. I must say we were very fortunate. Of course, we could have used more phenotypic assays. More phenotypic assay means more work, more funding, more people. And at that time, I left my job in Japan and I came to India. So we were in a little bit of difficult situation. But fortunately, we got the results that can be published. So the functional study of the lab-made genes, as we call them, can be done only when you overexpress them. Because if you knock out in a non-expressed piece of DNA, you do not know if something will happen in the cell. That would be very interesting. If somebody can make a library of, knockout library of the non-expressed genome. It has not been made so far to my best knowledge, but if one can make it and see the effect, that would also lead to discovery of new genes. The ENCODE project very clearly established, the encyclopedia of coding elements, that bulk of the genome is regulatory in nature. Tiny part is encoding proteome, and some of it is not coding. So all we are saying is how many non-expressing sequences can be encoded into functional genes and functional proteins. So now we have a mega challenge to, so we said we have been very lucky first time. Maybe next time we will not be lucky. So let us now make a database of intergenic sequences. Let us convert all of them into proteins, hypothetically, computationally in the computer. And we're talking of thousands of them, 2,500 or 3,000 from E. coli, whatever is documented, in E. coli, yeast, and Drosophila. We take it from the intergenic sequence database. We converted that into protein and peptides. And now we said, let us see what kind of phenotypic assay is available in our system. And then let us design protein based on the experimental validation that can be done in our lab. Okay. So now we started this large number of sequences and targeting that. So we found One sequence was very interesting coming from Drosophila, intergenic sequence. The sequences are totally different between the alpha defensin and Drosophila. Alpha defensin is an antimicrobial peptide which is commercially sold. So we found that sequences are totally different, but the structures are nearly similar, as you can see on the screen. So we said, now this is very interesting. Let's do an experiment and see what happens. So we synthesized the peptide, tested, and we found that it was really giving very strong growth inhibitory properties in several microbes. Then some of my students said, we are interested in malaria because there's a malaria lab that can validate our results. So we will target against MR1, EBA175, and MSP119. And nearly 2,000 yeast sequences, intergenic sequences were taken, and structures were predicted, and the rigid docking was done because we didn't have the computational power for flexible docking. So we said, whatever is available and possible with us, let's do that. And we self-learn. And then using certain tools, we could barely manage to do rigid docking. And we got some very interesting results computationally. And then we synthesized all the 3 peptides. And when we tested them experimentally, we had a big surprise that in presence of these peptides, not all of them together, but individually, up to 60% of parasites were prevented from entering RBCs. Now, this was again, it was an interesting result, not a very interesting result, an interesting result, which tells us, okay, you can go forward in the same direction. So your path is correct. And another big moment came when it was a totally non-linear, out-of-the-box thinking, that tRNA, as you all know, has been doing a social outreach program for millions of years. It has been taking the amino acid, bringing that to ribosome surface, latching onto another amino acid, and the polypeptide chain is made. It never expresses its own RNA into protein. It's only helping all the time. So we said, how about if we convert tRNA itself into a peptide? What will happen? Of course, there will be steric hindrances and other issues. We cannot do this in the cell. So we did a fairly large bioinformatics study, collected all the tRNA molecules, converted them into peptide sequence equivalents, and then we did some docking, some kind of bioinformatics, and then we selected the top one based on its stability and interaction pattern, and we synthesized it. Chemically got it synthesized, and the big surprise was that it kills leishmania. This was not a surprise. We were expecting something will happen. There was a bigger surprise. That IC value of 50, the 30 nanomolar, we see that it is killing the leishmania is getting, it is getting affected severely. So basically, we're killing the leishmania at such a low concentration. But the best part was that when you increase the peptide concentration even 10 times, human cells are not affected. Okay, so we are showing the TEM messages, the TEM images. As you can very clearly see that. Likewise, we found something more and something more. This is currently under validation. I think we have got the results already. We have to see how to publish it. And we also got Cas9-like molecules. We are now trying to hunt after more and more functional categories and look for collaborations. So this is the latest one we are after, but we don't have much results here. The work has just started. I'm hoping that, because, remember, we make peptide and proteins from the artificial gene made in the lab. Well, the same gene makes RNA first, and then the protein is made. So we cannot lose the RNA information. And from there, we want to create more RNA, as you know, in case of Moderna. Big news. So we said, why not look at the junk RNA artificially made in the lab, and will it have the potential to make more vaccines? Computationally, we have done this in certain set of sequences, and we've got very good results. We're looking for collaborations to do this experimentally. This work was picked up 2009. The first paper was out, and then I was invited by European Science Commission and the National Academy of Sciences and so on. It went on, so it is like a little comfortable to see that overseas people are liking the work. Of course, back home, nobody knows about our work, but overseas, people seem to be aware. Um, and we also wrote 2 textbooks, hoping that it will help the community. Our goal is to throw some light on the dark matter of the genome. There are 2 categories of sequences. One is strictly non-expressing, the other one is RNA expressing. So we have converted tRNA into tRNA encoded peptides. We are now, after long non-coding RNA, ribosomal RNA, and introns, we want to make peptides and proteins out of that, and fairly good amount of bioinformatics work has been done. And now, after maybe a couple of months, we'll start the experiments and look at the applications. This has led to spinning off the company with my postdoc, who's from the School of Life Sciences in JNU. So we both together set up the company in JNU called Foresight Biotech. And in future, we will be, looking at interacting with the investors. And we are actually talking to many people and hoping that this can be scaled up, because this is a new drug discovery platform. The best part is that you own the IP of every molecule that you make, because remember, nature did not make it in that particular organism. It has made in some other organism which has not been yet identified. We do not know that. But against all the existing data, such molecules do not exist. So this is starting from the junk DNA and then junk genes and junk RNA. So my students sometimes call it junkomics. They think I should book the domain name and start a website on this. We have recently entered into collaboration with the UK group, the Global Biofoundries, and in future we intend to. We are the first partners from India, and we are in future looking for some funding to provide these biofoundry services. Well, these are the people who were in the project right from the beginning, the RIKEN group, then the Singapore, Kerala, GNU, and now Triple IT. Well, there is always a big challenge. When you start something totally unexpected, you don't get results straight away, but we have stayed motivated with a big message from a small atom that says, be positive, even if you're always surrounded by negative things. So thank you very much for your kind attention, and I would be happy to take the questions here.
Thank you so much, sir, for your valuable insights. It was really a delightful experience to witness the journey of synthetic biology and all the factors that made the field what it is today. Your contribution to the field is just outstanding, and we really hope to take forward this legacy. I know a lot of you have some questions, but please hold on. We'll take them once the presentation by Team IGM ISO Pune is done. So now that I have talked about taking forward the legacy, I am delighted to tell you that today we have the members of Team IGM ISO Pune with us. They will be speaking on sustainable biomanufacturing of industrial compounds using a co-culture of cyanobacteria and E. coli. I request Sanjana from the team to go ahead.
Hello, am I audible?
Yes, Sanjana.
I'll just share my screen. Can you see my screen?
Yes, we can.
Okay, let me just start. Okay, so hello, I'm Sanjana from Team ISER Pune, and this year we're working on recycling atmospheric carbon dioxide for biomanufacturing using synthetic biology. Carbon dioxide levels are off the charts, and we must bring them down to minimize the worst effects of climate change. Annually, around 43 billion tons of carbon dioxide are emitted into the atmosphere. Fossil fuels contribute to over 74% of the global emissions, out of which 18% of the industrial emissions come from chemical and petrochemical industries. India itself contributes up to 7% of the global emissions. Presenting our solution. So, what do we want to achieve? We aim to create a carbon negative or carbon neutral model for industrial chemical synthesis, recycle atmospheric carbon dioxide to generate renewable microbial feedstocks, and convert these feedstocks into industrially relevant compounds. And how are we going to go about achieving this? We create a co-culture of cyanobacteria and E. coli to manufacture chemicals. Cyanobacteria fix atmospheric carbon dioxide as sucrose under osmotic stress, and we will modify it to secrete it in the culture medium. E. coli will then take up the sucrose and use it to manufacture chemicals. The chemical we've chosen for this particular project is butanol, but by substituting the strain of E. coli we use, we can manufacture a virtually endless number of products. Our choice of strain is superior to plants on many different fronts. It thrives at high light, temperature, and CO2 concentrations, and is thus suitable for outdoor cultivation. It fixes a much greater proportion of its carbon as sucrose, 90% compared to 15%. It is also less resource intensive to grow and can subsist on waste such as industrial flue gas and wastewater, so it does not compete with food production and agriculture. The co-culture facilitates a single-step conversion of atmospheric carbon dioxide into the desired end product, and this end product can be varied by changing the heterotrophic strain used. There is no sucrose wastage as it is directly consumed by the E. coli and does not have to be manually extracted or transported. The cyanobacteria also generate toxic reactive oxygen species. E. coli can quench these, leading to better cyanobacterial growth. So moving on to the wet lab component of our project, we begin by modifying our cyanobacteria. They naturally accumulate sucrose under osmotic stress, so our first challenge is to facilitate secretion. We will thus introduce a non-native sucrose transporter, CSCB. Additionally, to increase the yield of sucrose, we will overexpress SPS, the enzyme that catalyzes the rate-limiting step of sucrose synthesis in cyanobacteria. The second step involves modifying a butanol-producing strain of E. coli lent to us by Dr. Yazdani from ICGB to take up sucrose and break it down. Again, we start with the permease coded by CSCB, this time working in the opposite direction, and also add two additional genes, CSCA and CSCK, that incorporate all the carbon in sucrose into the native glycolytic pathways. As for novel contributions, S. elongatus UTEX 2973 is a relatively new chassis. Few parts have been well characterized in it. We aim to characterize promoters in this chassis for the first time. We begin by making a gene cassette comprising our promoter with a universal RBS, fluorescence reporter gene, and a terminator, flanked by sequences for genomic integration via homologous recombination. This insertion will be carried out using a CRISPR-Cas12a system. We plan to study promoter activity under different light and CO2 regimes by measuring the fluorescence of the reporter. We would like to thank Dr. Yazdani. His advice on shifting to a microaerobic environment for our co-culture helped us select optimal growth conditions for our microbes. We're also indebted to Dr. Anama, who helped us reevaluate our assays and offered insights into the butanol market. Now we come to the computational aspects of our project. Our main goals in the dry lab this year include optimal strain design, which involves identifying key gene deletion or overexpression targets that could increase sucrose yields in cyanobacteria and butanol yields in E. coli, identifying the important metabolic interactions between the two species in our co-culture, modeling the dynamics of the co-culture, while the biomass and key metabolite concentrations vary over time, and evaluating the impact of our biomanufacturing procedure on the environment. To identify optimal modifications, we use constraint-based modeling techniques such as flux balance analysis, which primarily make use of genome-scale metabolic models. These allow us to optimize a particular objective, say growth rate, by manipulating the fluxes to the organism's metabolic network. By adding genes as controls to the reactions, we can simulate the effects of gene overexpressions or deletions by controlling the flux through their associated reactions. This allows us to identify the optimal modifications to increase product yields. We extend FPA to a co-culture setting as follows. We assume that the two strains have an equal time average growth rate handled by Steadicom, and then look at the permissible variability in metabolic fluxes at different values of this growth rate using flux variability analysis. Analysis of the tolerated variability of each flux helps us assess the importance of the corresponding metabolic interactions. If we incorporate uptake kinetics of some of these crucial metabolites, we can predict the final yield and biomass values at different points in time through dynamic modeling. Finally, we would hope to plug our optimal strains into the dynamic model to see if the effects of the overexpressions and deletions are significant in the co-culture setting by comparing the butanol yield. Life cycle assessment, or LCA for short, is a technique to assess environmental impacts associated with all stages of a product's life from cradle to grave. It helps us conclude whether our project is carbon neutral, scalable, and economic. Our LCA extends from the bioreactor to the moment of delivery to a consumer while considering all the electricity consumed, carbon captured and emitted, and to some extent, costs. We'd like to acknowledge the help of Dr. Shirish Srivastava, who introduced us to constraint-based modeling through flux balance analysis, and Dr. Dwarak Ravikumar, who helped us understand what a life cycle assessment can add to our project. Now moving on to our human practices. On the collaborations front, we've worked with other cyanobacteria teams to organize a symposium to engage with each other and experts in the field. We're also working with some other teams to develop dynamic metabolic models and an international survey to assess people's opinions on GMOs. For outreach and education, we've planned an activity book for little kids to explain climate change, a comic book for teenagers to introduce them to the world of synthetic biology, and a YouTube series to explain the applications of synthetic biology in some detail. Our team has also created an informative high school survey, which will be followed by a set of webinars. Coming to expert interactions for future implementations, we spoke to Dr. Shantanu, who helped us understand the importance of reusing waste biomass to make our project environmentally sustainable. Dr. Malti, on the other hand, told us about the limitations of using biomass as animal feed. We also spoke to Dr. Anand, who helped us identify potential stakeholders and downstream processing strategies. Dr. Reena Pandit told us about parameters to consider for bioreactor design. To conclude, we would like to thank our PI, Thomas Pukudal, and our advisors, Prem, Viral, and Chinmay. We'd also like to thank our mentors, Rishik, Trisha, Mohit, and Mukundan. We'd finally like to thank all our sponsors. Thank you for listening. And that is the end of our presentation.
Thank you, Sanjana. I will now request Professor Dhar to give his insights on the project.
Well, thank you very much, Sanjana, and it was definitely a wonderful presentation of the work. And I know it's not very simple to make something consistently. You know, there is a difference between a microbe and microbial factory. Though we are trying to be, in our intent, in our practice, we're trying to think of microbe as a microbial factory. But what happens is that, and this is something that we observed, Shams was a PhD student in our school, so I know his work and his supervisor. They were working on making these recombinant proteins, but one of the issues they faced, which has been worked quite well by Professor K.J. Mukherjee, who was with us some time back, that whenever you're trying to make a product thinking that it's a microbial factory, somewhere down the line the cells become sick. And even if you have taken all the care, even if you have collaborated between, let's say, E. coli and cyanobacteria, and you think that it's going to happen because logically it makes sense, but in practical sense, something goes wrong, and the cells, when they make, let's say, biobutanol or any chemical for that matter, we are adding an additional metabolic burden on the cells. Even if your sink is good, even if it is removed from the cells, still for some time it is within. So cells try to get sick. So I would request you to please pay attention on the stress response elements to make sure that, it's like fooling the cells. We don't tell them that the product is being produced, so the cells will not get sick, and they keep on producing biobutanol. Many stress response elements have been identified. They have been up-regulated and down-regulated, different kind of elements. I remember there are more than 15 or 20 of them. If you want a list, I can share with you. It is very important while we are trying to convert a microbe into a factory, which is eventually our goal. And the nature of the factory is that the product must be consistently produced without the factory getting shut down. Because you're dealing with analog and nonlinear systems, it is very difficult, and the whole gene expression cassette is stochastic. It is very difficult to imagine that the cells will continue to produce the same product even if they do under the ideal conditions. So robustness might be a problem in future. So wonderful work, excellent thinking, and I think it's brilliantly supported by mentors both from academia and industry. You can't think of bigger names than the ones you have listed. They practically run the field. So I think it's in a good shape, in a good hands. So all the best and good luck to you.
Thank you so much.
Also, sir, some members from the iGEM team had some questions for you. So would you like to take those now?
Sure, please.
Thank you. Sanjana, Akash, Likhit, you guys can go forward.
So we were thinking about the benefits of immobilizing our cell factories here, E. coli and cyanobacteria, and would you recommend that we take that approach?
Well, you see, if you have to work on an industrially important chemical or a biochemical, you've got to use the same organisms, right? So we don't have an Indian model of microbial factory, though I've been insisting on that at many platforms. But with E. coli still is the best, yeast is the best, CHO cells, they are the best, and they are the only ones people are using. Though they are trying different model organisms, but it's very important to use industrially accepted organisms. So we are already there. From the organism perspective, I don't have any issue because you really cannot, you don't have more freedom. But my only concern is that how do you remove the product from the system as soon as it is produced so that the cell does not get overloaded, creating a sink outside the cell. And the metabolic engineering, community for nearly 10 years. I remember some work of people in MIT for nearly 10 years. It's like long back. It's not the recent 10 years. I'm talking of maybe 30 or 40 years back. So for nearly one decade, I think in 1980s, the approach was to overproduce at every step and think that by overproducing, by increasing the catalytic turnover, your final product will be more, right? Logically, it makes sense. Then they saw that, oh no, there are some more unidentified pathways where the flux moves to those directions. We have not identified them, so we're losing the flux. They try to plug that, okay, create a mutant, let it not go there. All kinds of things were done. Finally, they found, and this was shared with me by one of the very famous person from MIT who was a metabolic engineering expert. I'm blanking on the name. So finally, they said the solution was to produce the sink. so that you pull out the protein or the peptide or metabolite, whatever you are producing, you pull it out so that the cell does not react to its overpresence and it keeps producing. So that is where I would like to put my thoughts. And also because there are stress response issues, they come up over time. Even with the best lab, best efforts, even if you heard of repressilator, you heard of so many things, but repressilator or even the best design does not work for a long time. Even if, okay, it was an intent, it was well designed, and they did a great job by sensitizing the community and showing that. But we should never think of an engineering level, or you isolate the circuit from the environment. Go for cell-free systems, or go for a liposome-based transcriptional machinery, or maybe some vesicles, and try to see whether you are able to sustain the production of the chemical for a longer time. Again, more work needs to be done, and I know cell-free systems are very expensive, but that's what I would say. The direction is absolutely right. It is just that we do not know what's coming up, so we need to have alternatives.
So one of the things we were looking at to ensure that we could, in theory, make the co-culture more stable, because like you said, it could get sick, or one of the two components could get sick, was this idea of immobilizing the bacteria in these hydrogels, so that the E. coli would be within one hydrogel, the cyanobacteria within another. These would, or only one would be in a gel, and these would be positioned closer to each other. So in light of what you were saying about extraction, do you think that this is likely to make extraction easier? And in particular, we have this concern that E. coli is supposed to be taking up ROS species by cyanobacteria. So is that going to work if we put them in these separate cells like this?
Yeah, I mean, Akash, I'm sorry I don't have an answer to your question. I would have answered only when I work by my hands and I see what works. Logically, something might work, but in practical reality, something else might work. So co-cultures are definitely very attractive. There are people who are trying to, work with cyanobacteria. I know Himadri Pakrasi's work, who is trying to put the nitrogen fixation and the other pathway in the same bacteria so that you don't need fertilizers in future. So all kinds of things are happening. It is just that many combinations need to be worked out, and that's why many people like to go for directed evolution approach rather than the rational design approach. Because the rational design, you make 5 designs and you see that 5 of them are working in the lab for the next 1 month, and suddenly something happens which you didn't observe. So instead of that, what Jay Kiesling at Berkeley, when you're doing this artemisinin project, they create a large number of strains, large number of mutants, large number of strains. And then they keep on selecting that one after the other. And whatever survives at the end, that's the best design. Now, this is not an engineering approach, but this is a clever approach because it bypasses a lot of issues that you would come across. So I would say, I don't have a clear answer for your question. It is that your organism will eventually tell what strategy works best. But it's a clever approach, I agree.
So there's a question in the chat box for you.
Yeah, I can read it. So what's the evolutionary reason for non-coding part of tRNA to code for antimicrobial antidote, and how did you know that the express peptide would work as an antimicrobial agent? Okay. Evolutionary reasons we are exploring right now. We do not have a final answer. We have some pointers and will not be able to tell you that right now because the picture is not clear. The evolution of tRNA sequence versus its incorporation in the genome or its diversity. We're examining that question. But how did we know that leishmania is an anti-leishmania peptide? We did not know that. We had cancer cell lines, we had malaria strains, we had leishmania strains, the clinical strains. So whatever we had, we threw these tRNA peptides on all of them. And leishmania worked. So we have no idea that it is going to show such an effect. Yeah. But now that we have a result, we have made a library of tRNA peptides, and now we are examining that more systematically. But as you all know, a lot of money is involved. Students need to commit themselves. So we're looking for funding. We're looking for good students. Yes. Anything else?
There are no other questions, then I'll ask one related to our project. So, I mean, we've all been trying to incorporate sustainability into every aspect of our lives, given how things are right now. Um, so we thought we could do the same with our project, and we thought we could reuse our cyanobacterial biomass that we receive from the co-culture. So are there any ways that we could reuse the biomass? And because it's genetically modified and it has antibiotic resistance genes, does it pose any health risks while we're reusing it?
Yeah, the last part, you have touched a very important topic on the health risks and all that. Yes. You see, people have been working on biofuel for decades, and still you do not have a commercially accepted solution, because other solutions are becoming better and better. So therefore, but at the same time, it's a reasonable way to think that getting wasted, let's make use of that, whether it is sugar cane or whether it is something else that is a high sugar content or even a low sugar content, or it's part of the tree bark that gets wasted and all that. People have tried all kinds of things. And a pre-treatment in the industry costs a lot of money and a lot of effort to make it to the extent where you can hydrolyze it and get the best reaction. So really at the lab level, you can do a lot of combinations, no doubt about that. Metabolic pathway models are made, constraint-based models. First there was this ODE-based model, then people tried stochastic, then they tried the constraint-based models and the whole genome scale models. All the models are approximations, they don't tell you the exact, whatever happens in the cell, they will not tell you that. They will tell you what you have fed into the model. So many a times, I think most of the times, the experimental observation and the modeling outcome is almost always different, simply because we don't have all the components in our hand. So that is from the modeling perspective. But when you talk of taking the experimental results to the field conditions, that's where the trouble begins. Because the environmental people are after you, and then there are people who say there is health hazard. I should also quickly mention, I didn't want to say that, but since you also mentioned health thing, I was invited by the Organization for the Prohibition of Chemical Weapons, to give a talk. So it's like a parliament, you're addressing diplomats from 80 different countries. And they can shoot any question anytime, and you have to be absolutely ready. So one of the questions that came to me in the context of what you're asking was that, somebody asked me, you're making a lot of genes from the dark genome or the non-express genome. And can you make a lot of them? I said, yes, given funding, why not? Hundreds of them. And then the question came up, they said, oh, can you make a bioweapon out of it? And I said, this is something I've never heard, but people can think in different ways. So we were very careful that, we should not produce a viral protein, unintentionally. So like that, when you talk of health risks, definitely people will come up with a lot of questions, especially when you're trying to genetically modify the strain. Well, these are other problems. You see, as long as you're not releasing the strain in the environment, if you're doing in the lab, it is acceptable. But when it goes to the field conditions, So we have to be very, very thorough with that. But I don't think any health risk will be of a major concern, because you're not designing a bioweapon or something. You're simply using the organisms and, of course, doing some tweaking for the industrial, increase the industrial productivity. My major concern is not about that. My major concern is the sustainability of what you're producing. How long you can produce it, maybe 1 day, 2 days, 5 days, 10 days, and will it keep on, going forward, or will this whole system shut down at some time? That is where my thoughts are going, and that's why I think people have already thought. Akash was also saying the same thing. So you just have to do it, because what works in different labs. Different experiences, different labs, different culture conditions, different strains. So, we just have to do it. But at the same time, when you take it to the industry level, what you have learned in the lab may be totally different than what you experience in the industry. So that would be another level of optimization. But I would really encourage you to go along. Do not worry too much about the challenges. And whatever answer comes up, it's a new knowledge. You see, even the failures are inventions. You see what I'm trying to say. And for that matter, you have people from Reliance Group. Reliance story I know, because I was involved with them for some time. They're not too much worried about biobutanol. That's fine. If they get it, that's fine. But in the process, they will generate hundreds of patents. You see. So that's the value addition. That's what I would like you to think on, along with the final goal, of course. Yeah, I'm sorry, it's a long answer to this question. Yeah.
Thank you so much, sir. Next question is by Soham. Soham, would you like to read out your question?
So I will just make the question easier. So my question was basically the dark genome, the ternaming. It's too big to just randomly pick out some locuses and then see if it's making a functional protein. So how do you recognize possible loci? I can understand about the non-coding RNAs, which you can translate to produce proteins, but not about what about the locus which does not produce RNA at all?
Yeah, we have worked on the sequences the work I presented was about sequences that do not make RNA at all in the cell, the intergenic sequences. So the question is, if I can understand it correctly, now that you have a large possibilities, in fact, I can tell you, back of the envelope calculations, millions of new genes and proteins are possible just from E. coli. You don't need to go to Drosophila yeast or human. Imagine we are talking of human genome where you have 1.5% or maybe a little less or more protein coding. 98% or 98.5% is available for making new proteins and peptides. Okay, this is the scale at which we're looking. To handle this scale, you need a very robust bioinformatics pipeline, a robust filter, so now we have molecules. Suppose the intent is to make enzymes. Okay, we are now currently validating one prediction wherein an intergenic sequence is making a restriction enzyme in the computer model. It's easy to validate it. But if you're looking for lipases or you're looking for, maybe in the biofuel, the bigger problem is that of the enzyme. And that's why the industry is not able to take off because of the dependency of enzymes, which are very expensive. So depending on the phenotypic assay, we choose the target. If I'm looking for therapeutic peptide, I want to first of all know what is my target, what is the structure of the protein, against which now you have a large collection of molecules and do virtual screening and see which one fits in best, and then experimentally make those set of molecules, maybe top 5, top 10, and then go about it. If you're finding a weak correlation, then redesign the molecules and look for a stronger correlation. And that's the approach we are taking. We do not know beforehand what we're going to get. We just collect everything that is there and then do the modeling and the docking and so on.
Okay, so it's basically random. You would not target a specific loci if you have any idea?
We are targeting it, but later on. See, from the collection of the library, we are taking all the sequences, all the intergenic sequence of E. coli, all the intergenic sequence or tRNA or long non-coding RNA or pseudogenes or introns or whatever we are able to get. We are taking all of them. And now we have a large library. Now if the question is, give me an antimicrobial peptide. Give me an antimicrobial peptide against this particular organism, right? So you predefine the site. Now all we have to do is just take the structure of the protein and do a docking of the virtual screening, basically, of the molecule that are in the library. And eventually some molecule will show association and then redesign that to show a stronger association.
All right, thank you.
Ms. Sal, would you like to ask your question next?
Yeah, I'm not sure if this would be directly relevant to your expertise, but we were wondering what the best method in terms of sustainability and capital would be to actually extract bioethanol through our culture media. So we have been looking at a few methods per evaporation, adsorption, gas stripping, and so on. But we haven't been able to conclusively narrow it down.
So Misal, what's the question? Exactly the problem you're looking at, which you want to solve?
So the method of butanol production in terms of sustainability and cost, we want to pick an optimal method of extraction. And we were wondering if you had any inputs on that.
You see, if I was in your position, I wouldn't worry about the cost. I would first worry about the productivity. As long as I'm getting productivity and something is happening in the cell, I'm able to capture those molecules. That's what I would worry about. And then costing will be the problem given to the industry people to scale up. Because the moment you reduce cost in the lab, it doesn't scale up in the industry. They don't like our solutions. They have a different way of thinking altogether. So I would worry only about the productivity. As long as you can show the right numbers without the cell getting sick and you show a sustained production of this molecule, that's all you need to do. The rest of it should be taken by the industry. And you know, the thing is, it's not only the production of the, you know, the direction of the flux towards the production of biobutanol. That's one thing, but you also need to make sure that the butanol should come out of the cell. And what's the best way to do that? I really am not the best person, because this is those people who are in metabolic engineering field. They would be the right people to talk to, and I think Shams is there, so he would be knowing this. But I would really urge you to think of contacting the industry and asking their best practices, because they are making ethanol in large numbers. And what would work in this case, because whether you need to redesign the transporters, the membrane transporters, to make sure that, you know, we can transport it. But will that transporter work for a long time? I do not know. If that also starts, you know, toning down, so maybe you have to think of several solutions. Anything which is not about modifying the organism, as long as you can play with the culture conditions or creating an sink outside the organism, that would be most preferable, because that is sustainable. If that is not possible, then you need to probably play at the transporter level. But really, unless I work in the area, I will not be able to give you the right suggestion. I'm sorry.
Thank you. Next question is from a scientist journey. You have a rather interesting name and an interesting question. So would you like to ask your question directly?
Okay. Thank you for giving me this opportunity. I'm really glad to be speaking to such an amazing person. So my question is, even though we have all the current technology and we are trying out our best to bring biological machines into life, what is your prediction of how it would revolutionize the way we think about the industrial sector at the moment, and how it would come into mainstream? Like, when is a possible? What is the time scale at which when we can see this coming into the mainstream industrial sector?
What is coming to the mainstream industrial sector? May I ask you? I'm sorry, I didn't catch this
Yeah.
point.
I mean, like, moving on from the conventional industrial processes to the biological machines.
When you say biological machines, what is the example you have in mind? If I can request you to clarify it for me.
Um, so
Any
right
specific
now,
example, or is it a general aspect?
so in general, but like, I would like to know if you have any insights on the traditional infrastructural, I mean, industrial processes for making steel, or I mean, any of the other kind of hard industrial processes.
Okay, good. Because before I answer, I should understand the question. I'm sorry if I'm not been able to capture the essence of what you were asking.
Yes.
You see, in my view, and I would really urge all of you, if possible, to learn artificial intelligence tools, especially the metabolic engineering people who are looking for the right answer, but they do not have confidence in the advice that is given. So then that's where the AI tools help to a great extent. Again, they will not be replacement for the absolute solution, but at least they can enhance their accuracy of the answer. When you talk of biological machines, if you're talking of intracellular biological machines, if this is what you are asking me, then I would say that these will take time, because we have not solved the standards problem. We don't have biological standards, even if there is BioBrick and there is iGEM and all that, well, that's fine. It has its own purpose and role and exciting good students like all of you. But why BioBrick is not used by the industry? So we have to think of what are we dealing with? We are dealing with not an engineering system. We might impose the engineering constraints, but what we are dealing with is a system that keeps changing all the time. It's a 3 billion year old evolution that we are handling with few years of intelligence. How can we do this? So certain aspects might work, like people have tried to design a 6-base DNA. It is replicating, it's transcribing. Maybe they will face issues in the future. And the long DNA synthesis, for that matter, has been a mega hit. But when you talk of biological machines as an intracellular biological machines, I would say that it is going to take time. Don't worry about the hype that you hear, because we have not solved the problem of standards. You see, we are exactly the time when engineers were at the beginning of 20th century. There were hardly any standards. So here the standard development is all the more difficult. And this is also something I like to open up for discussion, maybe not today if you don't have time, but maybe you can think about it and discuss among yourselves. The kind of standard registry of standard parts that we are talking in terms of iGEM, well, first of all, it's not standard. We know this very well. Whatever works with the iGEM parts may again lose its robustness over a period of time. So instead of focusing on parts, and that's one thing I've been trying to tell people, instead of focusing on parts, please focus on modules. That means whatever is your system that you're trying to design, please see that the entire design itself should be like input output, a library of those input output values, the data sheet not on the parts, data sheet on the modules. Once you have a data sheet on the modules where you know that, okay, this is my input condition, this is the output condition, not of the parts, but the module, you will have a better design in the hand, because modules have more robustness than parts. When 2 or 3 parts come together, Remember, every part is nothing but a stochastic system. Now imagine, all of you know this already, I'm not saying something new. Imagine you combine two stochastic systems. Can you predict what will happen next? Nobody can predict. You combine one gene with another gene, another gene, another gene. You're combining several stochastic systems. How can you predict what's going to happen? That's why you lose robustness. That's why, the system fails over time, because it has adapted and evolved in a different way. So I would really urge you to think of creating a library or a standard registry of standard modules rather than standard parts. Because when you think of modules, so what is a module? For example, a lac operon is a module. For example, if you transfer the pathway to a mega-cloning vector, which has all the components, imagine you have one metabolic pathway, we are now talking of biobutanol. You have the entire biobutanol components on a vector. There are BGM vectors, mega-cloning vectors that are available. Suppose you transfer all of that to the vector, okay? So you have a cassette, you have plug in, plug out. So with this system, you can optimize it much better. You can put in the cassette in a vesicle and say, okay, the cellular elements will not interfere it. यहाँ हम cell free भी कर सकते हैं. So once you have a cassette in your hand, you can play it as an app. Exactly not an app, but in its concept as an app. And you can tone it down. You can, bring the numbers up and down because you have something in the hand that you can use entirely. But the moment you go to the genome level, one component interacts with 5 more components and you lose the accuracy. You cannot handle it because by the nature of system. So if you're talking about biological machines, delivery of the biological machines, people can use more terms, doesn't matter, but this will not happen in the near future. What will happen is that the technologies will come up. Like people are currently working on they're going far ahead in terms of the thought operating system. You heard of the operating system in computer, right? And earlier we used to speak something and write, and now you're typing, and somebody is using the mobile phone screen for typing, and somebody is speaking in the mobile phone or computer and it is getting typed. What next? Now people have started developing technologies that you can think and it gets typed. You can think and you send a WhatsApp message. People are working on that level. So that means they are bypassing all the other complexities, and they are, a thought operating system is already in place. Of course, not commercially available, but designs are already there. So you have to really think far ahead. So in terms of cells and a physical interface coming together to make something as a hybrid technology, this can happen much earlier. If you're combining cells with another surface and organic, electronics, that will come much faster than a machine that is embedded within the cell. I'm sorry, again, a long answer. Problem with the teacher.
Sorry, sir. Thank you for your insights first. But I would like to know about your insights in context to global warming at the moment, and how iGEM or your research could actually help reduce carbon emissions in the coming days. So how it would really how would it really translate to reducing carbon emissions from industry?
See, yes, thank you very much. Again, a very relevant point. The plain vanilla solution is plant more trees. But what will happen to research? We also have to work, right? I would say then make those cyanobacteria or plant species that can absorb more carbon dioxide. Because carbon dioxide being produced every moment, there is no solution to this. People have to make carbon dioxide because the industries have to function. Whether everything will switch on to the solar system, that's also a big question mark, because it might look very fancy to talk of solar cars or electrical battery cars and all that. But when you have to dispose of those batteries in the environment, it's going to be a bigger problem than carbon dioxide, global warming. Of course, we have to make sure that we are responsible, but at the same time, carbon dioxide absorbing organisms, if they are, again, there'll be a problem to release them. You see, my concern is not designing, but how do you, and that is where it was hinted that, the health impacts and other regulatory issues will come into picture. We just need to make sure that it is contained in a certain environment, but I think the focus, if you can convert waste water or the waste products into a recyclable material that people can use, including carbon dioxide, if that can be, such organisms can be designed, maybe in future we will get the regulatory approvals. But right now, people are concerned a lot about the environmental impact. But one has to keep working. Cyanobacteria in plants, I think, they're best materials for capturing carbon dioxide. Yeah.
Thank you, sir.
And also about, I think we have had a good discussion today. You all must be hungry and, to run for food. But I would also say that please do think about coming together. Now that we have 1,000 plus people interested in iGEM and synthetic biology, and the critical mass is already there. So please think of doing some kind of a national project or a national platform wherein different iGEM teams talk to each other and then in turn take the ownership of running the iGEM. I would not, personally, I'm not comfortable to use the word iGEM because, of different reasons, but let's make an Indian synthetic biology consortium or something like that. Yeah. And we need your support because students are the ones who will eventually run it. We are spent fuel, so thank you very much.
Thank you, sir. Thank you so much. With this, we come to an end of today's event. We hope you all took away some valuable insights. Big thank you to Professor Dhar for being kind enough to take out time from your busy schedule and join us for today's talk. We are really grateful for what we've learned today. I would also like to thank iGEM ISA Pune team for the fantastic presentation. Synthetic biology is a challenging yet promising field, and only by hard work and collaboration among students and researchers can we crack some of the world's most pressing issues. We hope that everyone present here today will take some valuable lessons back home and maybe work on it in the future and possibly create a difference in this world. Thank you, everyone. Stay safe.
Thank you very much, Vidisha, and thank you all for this wonderful gathering. So all the best and take care. Thank you.
Thank you, sir. Thank you so much.